Shedding or exfoliation is a physiological process for removal of deciduous teeth. This process requires the resorption of the supporting structure and the roots of the primary tooth, which eventually causes the shedding of the rest of the portion of the tooth. This place is then taken by the permanent tooth. Shedding is a complex process and is controlled by various mechanisms working together. Genetically programmed. As a person reaches a certain age, various mechanisms get activated, which gradually leads to the death of the formative cells and development of resorbing cells. Pressure from erupting permanent teeth. The pressure applied by the underlying erupting succedaneous tooth plays a vital role in exfoliation of deciduous tooth. But this pressure plays only a contributory role and is not a prerequisite for the exfoliation, as primary teeth without a permanent successor also eventually do resorb, although their exfoliation timing is later than usual. Pressure from increased masticatory forces. As a person grows, the muscles of mastication grow stronger. The increased masticatory forces are overwhelming for the small periodontium of the primary teeth and leads to its degeneration. Odontoclasts. These multinucleated giant cells are responsible for resorbing deciduous teeth. They are present in large numbers along the surface of the primary root nearer to the erupting permanent tooth. Microinflammations. On the root surface, the cementum forming cells cementoblasts are present, which must be damaged prior to resorption. Now, hormones and cytokines have no effect on the response of cementoblasts. Therefore, microinflammatory processes develop, which leads to their degeneration. It is the reduced enamel epithelium of the erupting permanent tooth which is known to release substances to initiate this process. Apoptosis. In multi-rooted teeth, many times the root resorption reaches the pulp chamber. Here, as dentine resorption continues, the odontoblastic cells have to be removed first. This is thought to be done by them undergoing programmed cell death. Pattern of tooth resorption. As we have read, the shedding of deciduous teeth is the result of progressive resorption of the roots and their supporting tissue. In general, the pressure generated by the growing and erupting permanent tooth dictates the pattern of deciduous tooth resorption. In case of anterior teeth, the developing permanent incisor and canine tooth germs develop in a position lingual to the deciduous tooth. From this position, as they erupt, they cause the resorption of the deciduous tooth root from the lingual side. Later, they occupy an apical position to the tooth and then they finally cause shedding. In case of deciduous molars, the permanent premolars start development between the roots of the deciduous molars. As the crown grows in size, the initial resorption takes place on the inner sides of the roots. However, as the jaw size is also simultaneously increasing, the deciduous tooth moves coronally such that the permanent tooth germ takes a position apical to the deciduous molar. As the tooth erupts, it again initiates root resorption from the root apex and finally leads to its exfoliation. The physiologic resorption of primary teeth is not a continuous process. Rather, the periods of resorption are interrupted by a periods of rest and repair, during which cementoblasts form calcified structures en route. However, the resorption process progresses faster than the repair and the primary teeth are eventually shed. Under histology of shedding, we have read about the increase in the inflammatory cells like neutrophils and lymphocytes. There is also degeneration of neuronal axons in the pulp, which in turn reduces its sensory function. Now we will study in detail about odontoclasts. These cells, responsible for removal of the dental hard tissues, are present in the resorption base. They are large cells with a smooth inactive side away from the dental hard tissue and a ruffled active side towards the dental hard tissue. In the cytoplasm, it has around 2 to 60 nuclei. It has numerous vacuoles and vesicles which are more concentrated towards the ruffled border. The cytoplasm is rich in mitochondria, sparse in rough endoplasmic reticulum but with extensive Golgi apparatus. Just peripheral to the ruffled border is a zone which has no cell organelles but is rich in filamentous proteins. This zone is called clear zone or sealing zone. Origin of odontoclasts. In the blood vessels, among all the white blood cells, It is the monocytes that are thought to be the precursors of odontoclasts. When required, monocytes from the blood vessels start migrating into the connective tissue and reach the site of dental tissue resorption. Here, they transform into mononucleated odontoclasts. Numerous such cells fuse together to form a large multinucleated odontoclast. On reaching the mineralized surface of the tooth, the odontoclasts get activated. Action of odontoclasts. The ruffled border has ATP proton pumps in its membrane, which releases protons across the membrane into the resorption bay, dropping the pH to 2.5 to 3. Moreover, the vesicles release numerous proteolytic enzymes. All these changes leads to the liberation of organic and inorganic particles from the tooth. These particles are endocytosed across the ruffled border and then packed into vacuoles, which are then passed across the smooth surface out of the odontoclasts.